Hi everybody, welcome to the 24 Hours of Stem Cells. My name is Adrian Teo and I lead the Stem Cells and Diabetes Laboratory at the Institute of Molecular and Cell Biology, IMCB A-STAR in Singapore. Today, I'm glad to share with you on our work relating to the use of patient-specific human-induced pluripotent stem cells for understanding diabetes disease mechanisms. So, data from the International Diabetes Federation has indicated that there are more than 400 million people in the world living with diabetes. You can also appreciate that majority of them are living in the Western Pacific region where I reside. In case you're not aware, every 6 seconds, one person dies from diabetes. And as of last year, more than $600 billion was spent on diabetes. What about the prevalence rate of diabetes? In the world, the world prevalence is at about 8 to 9%, whereas in Singapore, it is around 12 to 13%. That is way much higher than the world average. So that brings us to more than 500,000 people in the world living with diabetes. Now, because of this high prevalence rate in Singapore, the Ministry of Health in Singapore declared war on diabetes last year. It is a big problem here, and we purportedly have the second highest proportion of diabetics in the world. It is also claimed that 3 in 10 Singaporeans may have diabetes before turning 40, and we are always wondering how can we win the war against diabetes. So it's easy to think about diabetes, but what are the types of diabetes? Now you have the monogenic forms of diabetes, such as maturity onset diabetes of the young, which we call MODY, M-O-D-Y. It is caused by an autosomal dominant mutation in a gene that runs in a family. And then you can have neonatal diabetes, in which infants develop diabetes before the age of 6 months old, mitochondrial diabetes, or syndromes of insulin resistance. About 5 to 10% of the individuals can get type 1 diabetes. It occurs when the immune cells attack the pancreatic beta cells. Your beta cells die, they don't secrete insulin, therefore you get diabetes. Majority of the diabetic individuals are type 2s, in which the insulin resistance occurs in the classical organs such as muscle, fat, and the liver. The pancreatic beta cells try to compensate, but in the end they succumb, and you get type 2 diabetes. So last year, Jason Flanick published an excellent review in Nature Reviews Endocrinology. In the figure on the left, you see that they summarize all the known genes and gene variants that are associated with diabetes. Now the argument put forth is that we have learned a lot from the rare forms of diabetes, and we can make use of that information to learn more about the common forms of diabetes. For the figure on the right, you can appreciate that there are gene mutations present in this couple of genes, such as HNF1 beta, HNF4 alpha, HNF1 alpha, that gives rise to the severe form of MODY. Also, there are gene variants found in several of these genes here that are less severe, resulting in a type 2 subtype with a reduced beta cell function. So, we know that beta cells are not easily accessible from diabetic individuals, and the approach that we take is to use human iPS cells from these patients and differentiate them. In our group now, we typically derive human iPS cells either from the patient dermal fibroblasts or the peripheral blood mononuclear cells, and then use reprogramming methodology such as sender-viruses or episomal vector to make a human iPS cell that you can see on your right of the screen. A couple of years ago, we have already reported that it is possible to make iPS cells from diabetic individuals. On the top, you can see the bright field images of examples of human iPS cells from patients such as those with MODY 1, MODY 2, MODY 3, MODY 5, and MODY 8. Morphologically, they look like a control human embryonic stem cell. We also have iPS cells from type 1 and type 2 individuals that we are not showing here today. So what we did, of course, is to check for pluripotency by immunostaining, confirming that they are pluripotent, expressing OCT4, SOX2, NANOC, SSCA4, and TRA160. When these iPS cells are transplanted to immunocompromised mouse, they can make teratomas, having the 3 germ layers, the ectoderm, mesoderm, and definitive endoderm. And we also check that they are karyotypically normal. the major focus of my group is to then take these human iPS cells that we have made and differentiate them en masse into pancreatic beta cells to do in vitro disease modeling for diabetes. We're also looking at taking these type 2 human iPS cells to differentiate them into muscle, liver, fat, to look at mechanisms of insulin resistance, and also even diabetic complications, looking at neural cells, cardiomyocytes, or kidney cells. For the purpose of today's presentation, we're just going to talk about differentiation of these diabetic human iPS cells into pancreatic cells. So how do we do that in a dish? We typically have to learn from rodent development. And this schematic here, from the left to the right, you appreciate that we first have a mouse embryo that then develops going through the epiblast, differentiation towards definitive endoderm, primitive gut tube, foregut endoderm, pancreatic endoderm, endocrine progenitor, and ultimately to a pancreatic beta cell. We then try to map development in human against that of the mouse and try to do this in vitro differentiation. So to briefly summarize, we differentiate these human iPS cells stepwise towards the mesenderm, towards definitive endoderm, pancreatic progenitors, endocrine progenitors, and ultimately towards a pancreatic beta cell. So in this picture that I show you here, taken from a review that we published recently, it shows you that pancreatic beta cell differentiation is really highly complex and expensive. As we move the human pluripotent stem cells from the left all the way to pancreatic endocrine cells on the right, you can appreciate the green boxes, which are the signaling pathways that we have to activate during each step of differentiation, and correspondingly also the red boxes, which are the pathways that we have to inhibit in order to collectively push the cells towards pancreatic beta-like cells. It's highly complex and expensive. To give you a flavor of how it looks like, we start the differentiation from day 0, where you have little clusters of human iPS cells. These are in suspension cultures, and as differentiation progresses, we change the media every couple of days, and by the end of differentiation, we get these human islet organoid clusters in suspension that are human iPS-derived beta-like cells. So broadly speaking, we can use these human iPS-derived pancreatic beta cells to understand the biology of pancreatic beta cells in diabetes. For drug screening, when we have a large panel of beta cells from different donors, and eventually, hopefully, a cell replacement therapy. So following, I'm just going to be talking about an example of using an iPS cell to understand pancreatic beta cell biology in diabetes. So for this example, I'm going to talk about MODY5, maturity onset diabetes of the young 5, caused by an autosomal dominant mutation in this gene HNF1 beta, which is hepatocyte nuclear factor 1 beta. So why are we interested in this, and what is its importance? We know from the literature that HNF1 beta positive cells in the pancreas are multipotent. They can become the endocrine or the exocrine cells. Importantly, if you knock out both alleles of HNF1 beta in the mouse embryo and then rescue them via tetraploid complementation, you see strikingly that these mice exhibit pancreatic agenesis by embryonic day E13.5. Now, what is important between the mouse and the human for MODY is that many patients with HNF1 beta mutations only have the loss of one copy of HNF1 beta, and that's different from the mouse. Strikingly, these individuals also have partial pancreatic agenesis, and for us, we were fortunate to have access to MODY5 patients, in this case, those with the S148L mutation exhibiting dorsal pancreatic agenesis. So we worked with our clinician counterparts and recruited a trio from this family. You have patient number 6, the mother who is normal, the father, patient number 2, who has the mutation and has diabetes, and then the daughter who has the mutation but not developed diabetes yet. The approach and strategy would then be to make at least 3 iPS cells from each of these individuals, and then check the absence or the presence of the gene mutation as a first cut. We have not showed you here, but essentially we have a pancreatic differentiation protocol in which HNF1 beta gene expression peaks at about day 10 of our differentiation. On day 10, we took our control cell lines and performed microanalysis. In this heat map here, I'm showing you on day 10 that many of the pancreatic markers are beginning to be expressed, such as PDX1, HNF1 beta, PAX6, GATA4, GATA6, and PTF1A, amongst many others. On day 12, we also confirmed the percentage of HNF1 beta positive cells in our dish, and in this FACS analysis, we show you that there is about 92% of HNF1 beta positive cells. So we have made the iPS cells. We have a protocol to study HNF1 beta biology using these modified iPS cells. The next thing that we then did was to take many of these iPS cells lines and then differentiate them towards pancreatic progenitors and ask the question, what is happening in these cells with HNF1 beta mutation? If you look at the legend on the right, you have in brown non-family control, in red the family control, which is the mother, and then those two in orange, which is the father and the daughter, both with the mutation. Here I'm showing you some qPCR data carried out in time-course fashion. On the x-axis, you have day 0, 12, 14, and 17 of differentiation. And here we look at 5 genes, HNF1 beta, PDX1, GATA4, PTF1A, and GATA6. Now let us just focus on the orange bars, which represent the individuals with the HNF1 beta mutation. Immediately you will realize that it's striking that there appears to be a compensatory increase in the gene expression of HNF1 beta, PDX1, GATA4, and GATA6, but no change in PTF1A. Now these 5 genes are known to cause some form of pancreatic agenesis, and immediately it tells us that in MODY5, there is no decrease in PDX1, GATA4, or GATA6, and so mechanistically, this HNF1 beta autosomal dominant mutation is independent of these other genes that I'm showing you here. We then confirm this compensatory increase at the protein level by immunostaining, and here you can see that individuals with a mutation have higher levels of HNF1 beta and PDX1. We also carried out FACS analysis on PDX1, and again we see that there's high level of PDX1 protein in the mutants as compared to the control. We next wanted to study this relationship between HNF1 beta and PDX1 more closely. So we performed some luciferase assays. Here we have 2 PDX1 promoters, one a 7 kb full-length promoter, and the other one, which is a shorter form of PDX1 promoter, containing the areas 1 to 3, the autoregulatory region of PDX1 promoter. When we transfected day 12 cells with the PDX1 promoter, we see that PDX1 promoter is active, and strikingly, when we overexpress HNF1 beta, there is a suppression of PDX1 transcriptional activity. Now, HNF1 beta usually forms a dimer. It can be a homodimer with itself, or as a heterodimer with HNF1 alpha. So we wondered if HNF1 alpha was doing anything in this case. So we repeated the luciferase assays now with either 1 alpha or 1 beta or in combination. Here again, PDX1 promoter is active. When you overexpress HNF1 beta, it suppresses the PDX1 transcriptional activity. When you overexpress HNF1 alpha, it really doesn't do anything, telling us that the suppression of PDX1 transcription activity at this stage of differentiation is solely dependent on HNF1 beta. Now, if we think back at the transcript profile of HNF1 beta and PDX1, it then becomes not too surprising because HNF1 beta transcript needs to decrease because the transcript expression of PDX1 starts to increase. So we were really interested to look for genes that were downregulated in this disease modeling of MODY5, and amongst many genes that we looked at, the only one that we saw downregulated was PAX6. This is the expression profile of PAX6 during differentiation. It looks pretty similar to HNF1 beta, except that it's a bit more right-shifted. We also performed FACS analysis for PAX6 and HNF1 beta on our day 12 cells, And we found that there are 22% of cells that are double positive for PAX6 and HNF1 beta. So what is PAX6 and why is it important? So PAX6 has been reported by Mikey Sander that it is expressed as early as embryonic day 9 to 9.5 during pancreatic progenitor formation in cells exclusively committed to the endocrine cell fate. PAX6 deficient pancreatic progenitors have been shown to be unable to mature and reach terminal differentiation. PAX6 has been reported to directly regulate beta cell function genes. And very importantly, individuals with PAX6 heterozygous mutation actually do exhibit glucose intolerance. So it was very nice for us to work with the clinician collaborators because we were now able to go back to our modified individuals and perform more clinical characterization. And in this table here, you can appreciate those numbers in red, that our two patients with mutation appear to have higher levels of fasting glucose, lower levels of fasting insulin, and strikingly, the plasma glucagon levels are a lot higher than the controls. So currently, this is our working model. We started with modified human iPS cells harboring the mutant HNF1 beta gene. For the data that I have not shown you today, but published in stem cell reports last year, we saw an early compensatory increase in a couple of endoderm genes. Subsequently, there was a compensatory increase in HNF1 beta, PDX1, GATA4, GATA6, and so on. And that's why we think that there's a low occurrence of neonatal diabetes despite pancreatic agenesis. Amongst many genes that we looked at, the striking gene that we found to be decreased was PAX6. And given the information I told you about PAX6, we think that it highly contributes to beta cell dysfunction in MODY5. So with this example, I'd like to re-emphasize our proposition for using the value of using these diabetic iPS cells, differentiating them to pancreatic cells to understand diabetes disease mechanism, because many type 2 diabetes gene variants increase risk through pancreatic beta cell dysfunction, that there are strong mechanistic links between type 2 diabetes and well understood monogenic forms of diabetes, and has been reported that the type 2 diabetes associated gene variants show the strongest localization to islet enhancers. So with that, I'd just like to introduce my team, the AT lab members. We have Natasha, a postdoc, Hui Hui and Elvin, my research officers, and then we have Larry, Lynn, Blaze and Sabrina, my PhD students, and Daniel, an undergrad working in the lab. So with that, I'd like to thank you for attending the session today, and hopefully we can all beat diabetes together using iPS cells to understand diabetes disease mechanisms, using it for drug screening, and hopefully for cell replacement therapy. Thank you very much for your time.